Good morning. Welcome to Photosynthesis Part 2. So we begin with our topic, process of photosynthesis. Photosynthesis is a process which converts light energy to chemical energy. And green plants trap light energy of the sun and change it into chemical energy which is trapped in the form of carbohydrates. Now, so the process of photosynthesis involves two phases such as light dependent phase and light independent phase. Light dependent phase can also be called as photochemical phase and light independent phase is called as dark phase. So what is this light dependent phase? In this phase, it is very sensitive towards light. Light initiates a series of reactions which converts light into chemical energy. And as these reactions can occur only in the presence of sunlight, this phase is also called as photochemical phase. The other one, which is called light independent phase or a dark phase, is a phase which does not require sunlight and chlorophyll. The reactions utilize chemical energy stored during the light phase and leads to the formation of carbohydrates. So, these reactions are also known as Blackman's reaction, and this phase is also called as biosynthetic phase because it can synthesize carbohydrates in the absence of light. Now, it should be clearly understood that both the phases, photochemical and biosynthetic, occur simultaneously. Now, let us see this light dependent phase in detail. Light reaction takes place in the thylakoids of chloroplast during daytime. If you see a diagram of chloroplast, you can clearly see the thylakoids, and so this light reaction takes place in the thylakoids of a chloroplast during daytime. And light reactions consist of the following steps, such as first step is activation of chlorophyll. Here what happens, chloroplasts absorb sunlight and chlorophyll A and B molecules get activated by absorbing photons. Second step is photolysis of water. In this process, it absorbs sunlight, splits water molecules into hydrogen ions and hydroxyl ions and releases electrons. This phenomenon of splitting of water into two compounds under the influence of light energy is called photolysis. Photo means light, lysis means breaking up of water. Third step is evolution of molecular oxygen. The hydroxyl ions produced by the photolysis of water combine together under the action of an enzyme called catalyst or catalase. The reaction results in the formation of water molecules and release of molecular oxygen. Our next step is formation of NADPH, whose full form is nicotinamide adenine dinucleotide phosphate. So you have to mug it up, this full form of NADPH. And what happens here? Hydrogen ions released during photolysis of water is picked up by the acceptor molecule NADP. This NADP gets activated and reduced to form NADPH. Our last step is photophosphorylation. In this process, ATP is formed. Here, the energy liberated in the form of electrons during the photolysis of water is absorbed by a low energy compound called ADP, and its full form is adenosine diphosphate. ADP combines with one inorganic phosphate and gets converted to high energy compound, that is ATP. So here ADP is converted to ATP, and since the energy of this reaction comes from the photon, the process is called as photophosphorylation. So these are the steps which are carried out in light dependent phase. Now when we see the light independent phase, what happens? What are the process or the steps taking place here? Light independent phase is also called as a biosynthetic phase, and it involves reduction of carbon dioxide to sugar and formation of carbohydrate in the stroma of chloroplast. See the difference. Light dependent was carried in the thylakoids, whereas light independent is carried out in the stroma of chloroplast. And this phase does not require light and it is thus called light independent phase. The biosynthetic phase of carbohydrate is also known as carbon assimilation. This process requires sufficient energy which is provided by ATP and NADPH formed during the light phase. And this both the phases are taking place simultaneously. So, the fixing of carbon dioxide by the chloroplast and the synthesis of carbohydrate takes place in a series of steps, each controlled by a specific enzyme. This cyclic reaction is called Calvin cycle, and this is the American chemist who investigated these reactions. So, overall equation of photosynthesis you might have mugged up. So, you can go through that and how this light dependent phase and independent phases are carried out. Please be clear for this following steps. Factors affecting photosynthesis. The process of photosynthesis is influenced by both environmental and internal factors. Environmental factors here refers to external factors. So, let us see in detail about the external factors. The first factor is our light. Average light intensity and long duration of light conditions are sufficient for normal photosynthesis, and rate of photosynthesis is very slow at low intensity of light, that is at the time of dawn and dusk. So, higher light intensity increases the rate of photosynthesis up to a certain limit, after which it becomes constant or has inhibitory effect. And maximum photosynthesis occurs in blue and red light. The rate of photosynthesis increases with the increase of carbon dioxide concentration and vice versa. Higher the concentration of carbon dioxide inhibits the photosynthesis. The optimum range of temperature for photosynthesis is 24 degrees centigrade to 35 degrees centigrade. The rate of photosynthesis increases with the rise in temperature, and the rate of photosynthesis decreases beyond the maximum optimum temperature. However, the range of temperature required for photosynthesis varies in different species of plants. Only 1% of water absorbed by the plant is utilized for photosynthesis. The shortage of water in plants because of reduced absorption from the soil or excessive transpiration results in loss of water or water stress in the plant, and this causes the closure of stomata and thus decreases the diffusion of carbon dioxide in the plants, resulting in lower rate of photosynthesis. Nitrogen is the basic constituent of chlorophyll and the enzymes involved in the process of photosynthesis. Reduction in nitrogen supply thus has an adverse effect on the rate of photosynthesis. It said that photosynthesis is not affected by all the external factors or environmental factors at a given time. In 1905, Blackman postulated the law of limiting factors, and this states that when a process is conditioned or governed by a number of different factors, the rate of process is limited by pace or of the slowest factor or the factor present in the least amount. So, in other words, we can also say if all the other factors are kept constant, the factor affecting the rate is at minimum, and the rate of the process does not increase further even though the amount of this factor is increased, as now another factor has become a limiting factor being in at least amount. And the rate of process is therefore now dependent on the second factor available in the smallest amount. So, take an example. Let us say that a plant is kept in a condition where carbon dioxide is available in plenty, temperature is high, but light is limited due to cloudy weather. So, what will happen? The rate of photosynthesis under such situation will be controlled by light intensity, and if the light intensity is increased, the rate of photosynthesis will rise till a maximum level is reached. Hence, here what happens? Carbon dioxide becomes a limiting factor, and thus the rate does not increase further. Now, if we see the rate of photosynthesis is dependent on concentration of carbon dioxide, at this stage, rise in carbon dioxide concentration will increase the rate of photosynthesis. Now, further increase of carbon dioxide concentration will not influence the rate of photosynthesis. Now, the light becomes the limiting factor. Like this way, the Blackman has postulated its limiting factors. So, it said that photosynthesis is never affected or not affected by all the environmental factors. Now let us welcome to internal factors. There are certain internal factors also. So first of it is leaf factor. So first factor is our leaf factor. Leaf age, leaf size, leaf angle and leaf orientation are important leaf factors which influence the rate of photosynthesis. Secondly, rate of photosynthesis increases with the age of leaf till the leaf is young and fully mature. With increase in age, leaf loses chlorophyll resulting in a reduced rate of photosynthesis. The thickness of cuticle and the distribution of number of stomata also affect the amount of light and carbon dioxide entering the leaf and thus influences the rate of photosynthesis. Chlorophyll content. The rate of photosynthesis increases with the increase in chlorophyll content because the more solar energy trapped for this process. Next is our translocation. The food formed during photosynthesis translocates to different parts of the plant through phloem where it may be converted into starch and gets stored. And the non-translocation or accumulation of carbohydrate or end products of photosynthesis in the cells reduce the rate of photosynthesis. Protoplasmic factors. The process of photosynthesis stops and comes to a standstill in the complete absence of oxygen. The potassium ions are the important catalysts which are required during the manufacture of food in the plants, and the process of photosynthesis does not take place in the absence of potassium. So it said that deficiency of water in the protoplasm closes the stomata of leaves and reduces the rate of photosynthesis. Experiments on photosynthesis. A number of experiments can be performed to understand the condition, requirements and end products of photosynthesis. So a few we will explain. So light, carbon dioxide and chlorophyll are the essential factors for photosynthesis, and oxygen is evolved during photosynthesis and starch is formed as a product during photosynthesis. But before we describe these experiments, let us understand the method of testing the presence of starch in a leaf. Experiments on photosynthesis. Before conducting the experiments, we have to do a procedure for the experiments to be carried out. So the procedure is destarching. So what is destarching? It is the removal of any pre-existing starch from the plant. A plant used for experiments on photosynthesis need to be destarched, which can be done by the following methods. So what are those? First method is keep the potted plant in dark for 2 to 3 days. And secondly, wrap the leaves of a plant in an aluminium foil 2 to 3 days to avoid any exposure to light. And during this period, all the starch will be removed from the leaves. Secondly, we have to conduct the iodine test, and this is a test for starch. So here what to do? The leaf should be tested for the presence of starch before the start of an experiment. So for the this iodine test, detach the leaf from a destarched plant and dip in boiling water for a minute to kill the cells. Now boil the leaf in methylated spirit over a water bath till it becomes pale white because of loss of chlorophyll. Then third step is dip the leaf once again in boiling water. It will soften the leaves and that has already become brittle because of alcoholic dip. Last is spread the leaf in a petri dish and pour a few drops of iodine over it. The leaf will turn blue-black if starch is present and the stains brown if the starch is absent. So that's it for today. So next class we'll see the experiments on different factors. So here we are winding up with the procedure of destarching and test for leaf, that is iodine test. Thank you and have a good day.